So pulmonary fibrosis is a disease involved in excess scar in the lung, and it's the end stage of a lot of lung disease. And this disease involved the accumulation and proliferation of the myofibroblasts in the lung. We can see that. Yeah. Can we back?
Let's try it again. I'll restart your timer.
Okay. So the myofibroblast differentiation is regulated by a lot of enzymes. These enzymes requires metabolites and cofactors. And we are interested in two of these enzymes. One is the PAP1 and the other one is the HIF1 alpha. And these two enzymes implicated in activation of myofibroblast differentiation and pulmonary fibrosis. And these enzymes are also regulated by metabolites and cofactors. So the PAP1 PAP1 catalyzes poly-ADP ribosylation, and it requires NAD+ as a cofactor for its enzymatic activity. And the cellular NAD+ level would affect PAP1 activity and potentially the myofibroblast differentiation. In the meantime, consumption of the NAD+ by PAP1 activation would affect other NAD+ dependent enzymes, such as SIRT1, which are known to regulate cell fate and myofibroblast differentiation. The question is there a role for metabolic alternation in the regulation of myofibroblast differentiation? So we isolated fibroblasts from IPF patient or fibroblasts from the control subject and compared the PAP1 expression and activity in these cells. The result showed that the PAP1 expression is highly induced in fibroblasts isolated from IPF patient. And the activity is also highly induced and much higher in the cell from IPF patient. This is correlated with the induction of alpha smooth muscle actin, which is a marker for myofibroblast differentiation. The actin expression is much higher in the fibroblasts isolated from IPF patient. So this correlation suggests that PAP1 regulation is important for myofibroblast differentiation. We know that PAP1 uses the NAD+ as a substrate to catalyze the extension of poly-ADP ribosylation. And this modification of the protein is most of them are transacting factors, would potentially affect the myofibroblast differentiation. So we analyzed the potential metabolic change in the fibroblasts isolated from IPF patient and the fibroblasts from the control subject. This heat map showed the heat map generated according to the concentration of the glycolysis metabolites and cofactors and in these cells showed that the glycolysis metabolites are increased in the fibroblasts isolated from IPF patient. The green color stands for low concentration, and the red color stands for high concentration of these metabolites. So this suggests that the differentiated cell seems to be metabolic more active TGF-beta is an important inducer for myofibroblast differentiation. To confirm that the differentiated cell are more metabolic active, so we treat the human non-fibroblasts with TGF-beta for 48 hours and then analyze the metabolites by LCMS and GCMS. And the same, the heat map shows according to the concentration of the metabolites in the cells, indicates that the TGF-beta treated sample has much higher concentration of the glycolysis metabolites in these cells. So this is summarized in this figure. The darker color indicates increased metabolites in the differentiated cell, such as succinate, fumarate, malate, oxyacetate, etc. And the statistical analysis showed that the cofactors, which shown in red, ADP, AMP, NAD, MP, NADPH, FAD, etc., are significantly increased in the differentiated cell. And the ATP and NADH are significantly decreased in the cell from IPF patient and TGF-beta treated fibroblast. And the other metabolites such as MAR, F6P, alpha Kg, OAA, etc., are increased in the differentiated cell, which are cells from IPF patient and TGF-beta treated cells. To confirm the metabolomic analysis result, we also did a colorimetric assay to test the most changed ATP and ADP cofactors. The result is consistent with metabolomic analysis, that the ATP are decreased in both cells from IPF patient and TGF-beta treated cells. And the ADP instead increased in these cells. So the question is, this study suggests that the differentiated cell are more metabolic active. So, and there is a metabolic alternation during the differentiation. So the question is, are these metabolic alternation important for the myofibroblast differentiation? HIF1 alpha is another important regulator for myofibroblast differentiation and fibrosis. It activates the myofibroblast differentiation and promotes the fibrosis. It's also induced by hypoxia. And the prolyl hydroxylase domain protein, PHD, were ubiquitin, were marked for ubiquitin and proteasome degradation. And this enzyme were prolyl hydroxylate the HIF1 alpha. And it requires alpha Kg, alpha ketoglutarate, as a substrate for the modification of HIF1 alpha. And this alpha Kg is a TCA cycle-metabolic intermediate, and which are significantly changed during the differentiation. So we wonder if the alpha Kg level would regulate myofibroblast differentiation. So the octanol alpha ketoglutarate is a cell-permeable alpha Kg analog. So we treated the human non-fibroblasts with this analog and tested the alpha smooth mass actin gene expression. The alpha smooth mass actin is the most important marker for myofibroblast differentiation. So the result indicated here suggests that the dose-dependent suppression of alpha smooth mass actin gene expression is correlated with myofibroblast differentiation. So the alpha ketoglutamine would suppress the myofibroblast differentiation. So to summarize, we found that the fibroblast isolated from IPF patients is more metabolically active than the cell from the control subject. And the TGF-beta can induce the myofibroblast differentiation, and it also induces the metabolic activation. And there is a role for the metabolic activity for myofibroblast differentiation. Supplementation of the cellular alpha Kg would suppress the alpha smooth muscle actin gene expression and myofibroblast differentiation. So we'd like to thank the member from PanLab, and this study was supported by a grant from NIH and utilized the Metabolic core service from University of Michigan. And we'd like to thank them for their excellent support and services. And the human non-fibroblast was get from University of Minnesota from Dr. Craig Hank. Yeah.
Questions? Yes.
So I was curious about your octyl alpha ketoglutarate. So that is has been shown by a few labs now to go in so instantly that it's really the concentration internally within an hour is very high, and you get almost an instant sort of toxic effect. Rather, people have been moving to dimethyl alpha ketoglutarate. Have you tried that?
Yeah, we didn't observe the significant toxic effect for that. The cell we are using is human non-fibroblast. They can survive very well. So we didn't, maybe potentially we can do apoptotic assay to check that, but we didn't, just from the appearance and the morphology, we didn't see any significant toxicity.
Thank you for a very interesting talk. My question, how was extracted fibroblasts? Because if ADP levels are higher than ATP, it means that something wrong with extraction. Could you comment on that? Mm-hmm.
Yeah, that's a very interesting question. I think we also noticed that I think one of the reasons is ATP low and ATP high is during the differentiation, they produce a lot of protein. It uses a lot of energy to synthesize these proteins. So that consumed the ATP. And in the meantime, it's more metabolic active, and it needs a lot, generates a lot of ATP. And another side is it uses energy to differentiate and to produce the proteins such as alpha smooth muscle actin. So it's a balance. So the time point is very important. So now we are checking the 14 days after, 48 hours after the TGF-beta treatment. We are planning to do another early time to see if there is any difference. Yeah, so when the ATP was used, it will generate ADP. Yeah.
One last question.
I'm very interested in the ester of the alpha ketoglutarate. Somebody had shown about 10 years ago that you could prevent liver cancer by administering this agent. Did you show that it was actually hydrolyzed inside the cells, and what sort of concentrations of alpha ketoglutarate were you able to get in your system? Did you measure that?
We are mainly focused on the glycolysis metabolites, so we didn't measure that. Yeah.